Dear students, good afternoon to you. I'm really very happy to discuss you about soil and plant analysis. The learning objective are to understand the meaning of soil and plant analysis testing, explain the different processes in soil and plant chemical analysis, processing of soil and plant samples and their chemical analysis. So fundamentally, this lecture is not exactly lecture, it is a kind of practical. In practical, I made a promise to you that I will teach you soil sampling and plant sampling, particularly processing of plant and soil sampling. Processing here means taking a sample and making it ready for chemical analysis. Whether it is soil sample or plant sample, it is required to be processed. It is required to be made a representative sample in the best possible way. In both the cases, whether it is soil sampling or plant sampling, they are they have to be representative samples. So we start with soil testing. So actually, this practical will supplement or complement the lecture on soil fertility evaluation. If you remember, we had a lecture on soil fertility evaluation where we have seen that nutrient deficiency symptoms we can diagnose. We can diagnose the problem of soil fertility by quality analysis, quantitative analysis, that is soil testing or soil analysis. And then in the end, we have seen by doing some experiments and also by some remote sensing techniques. So there were a number of ways by which we could evaluate the soil fertility. And in those cases, you have seen that quantitative analysis of nutrients in soil as well as in plant is also an important part of soil fertility evaluation. Now let us start with soil testing. What is soil testing or soil chemical analysis? So soil test may refer to soil analysis for one or several possible reasons, maybe for available nutrient, total nutrient in soil, in order to determine the fertilizer recommendation. So this could be one purpose to develop the fertilizer recommendations. We need to go for soil testing. And what are the different components or steps in soil testing? It involves collecting of soil samples, preparation for analysis, that is processing of soil sample, and then chemical or physical analysis, whatever is your aim. Then after the analysis work, you need to work out or you need to know what does it mean? Means interpretation of analysis results, and finally give the recommendations of the fertilizers or whatever is your requirement or purpose of soil sampling. The steps are soil sampling, preparation, and then extraction or actual analysis of the soil samples, and then interpretation of soil analysis data. So there are 4 steps in this. First step is soil sampling. It has to be representative sample because sometimes you use just 0.5 gram, 1 gram, even 0.1 gram of the soil sample. Out of 1 hectare or 2 hectare, if you have just 1 or 2 sample, then if it is not true representative of that large area, then you will get erroneous results and erroneous recommendations. So that way you will not get good growth and yield and economics of the crop for which nutrient recommendations have been developed. So it has to be representative sample of the whole field. And of course, the number would depend upon variability and size of the field. Normally, higher the variability, more is the sample number. Level, land, uniform soil, 1 sample per 4 to 5 hectare. Only 1 sample is enough for 4 to 5 hectare if your land is uniform and land is very well leveled. Otherwise, you need to increase the number of samples. Undulating topography, bottomlands, uplands, and slopes have to be sampled separately. If topography is uneven, then from every section you need to collect the soil sample. And normally one sample for less than a hectare area. Best time for sampling is before sowing or planting. So that is the best time, means when you have not plowed the land. It has to be taken from undisturbed land. If you have plowed the land, then sampling should not be taken. So suppose you have harvested the crop and you want to do sowing of the next crop. So immediately after harvest of the crop, you should take the sample from the undisturbed soil. Or what can be done if you do not have time, say 8 or 10 days before harvesting of the crop, one can take sample, because at that stage crop will not take any nutrient from the soil. So that is also a time. So, but never, it should never be taken from plowed fields. So each sample should have a label. You need to put certain marks, certain paper, you can write on a paper, and that paper should be attached to the sample. That is known as a label. Describing field number or field identification, farmer's name, address, previous crop grown, and crop for which nutrient recommendations are sought. Now, how to take it, how to actually do it. So for general advisory, general purpose, you know that samples are taken with a depth of 0 to 15 centimeter, which is also called as plow layer. Sometimes it may be 30 centimeter if you have deep-rooted crops. In pedological studies, certain studies, even deeper sample is required, say 180 centimeter. And if it is done for fruit crops or crops which are very tall and their roots can go deeper, you can take much deeper sample also. So it depends upon nature of crops. But for our purpose, field crops, agronomic crops, 0 to 15 centimeter is ideal. While taking soil sample, we are sampling a soil volume, not the area. So volume of the soil is important because volume is equal to depth into area. The number of cores for composite sample depend on variability of the soil fertility. Here, dear students, number of cores means the places, how many places you are going to take the sample. Actually, cores here is representing the sampling. Cores, sometimes you get a kind of core sampler, C-O-R-E. Core sampler is a sampling tool that takes the sample from the soil. So here sample cores means the number of samples. The number of samples for composite sample depend on variability. If variability high, then you need to take samples from many places and convert it into one. That is the mean. So in practice, 20 to 30 cores will generally serve the purpose. Means from 20 to 30 different places it should be taken, then you can make one out of these 20 to 30 cores. Selecting the points where this core sampling or from where we should take the sample. So selecting the point, there are different patterns. Somebody can go zigzag path in the field, avoid 50 centimeter border of the plot because border area may have different fertility than the main field. So border should be avoided. All cores should sample equal volume and weight. So all cores here means all sampling or subsampling from different areas of the field should have the equal volume and weight. These are collected in a bucket or a container. Means every time you take a sample from the soil, you need to put it into the bucket and make a composite sample out of 20 to 30 cores or 20 to 30 places. These are collected in a bucket, soil thoroughly mixed. Then you have a large sample size. You need to reduce it and you need to ultimately have it 500 grams. So you can thoroughly mix it and about half 1 kg can be taken. So use polythene bag or paper carton and label. Do not use bags previously used for fertilizer or lime. Care should be taken. This is how one can take sample. So this is a field. Random sampling can be done this way. And it is certain points can be made like this. And one can walk in the zigzag fashion to collect the samples. If it is very, very large field, then it can be divided into different parts. In 1 part, 2 part, 3 part like this. In every part, you can take from 5 places and like this. So there are ways and means are available to take the representative sample or to decide the place from where sampling should be done. Now, what are different tools used? So there are different tools, number of tools depending upon requirement. A cutaway soil sampling tube and the spade are the most common. Cutaway means initially it is a tube, and then when you progress after 10 centimeter, 15 centimeter, and then you cut it, and it is not complete tube, it is just half portion is cut. I will show you now this kind of auger. Post hole and screw auger is also employed. Post hole auger is for say deeper samples, much deeper samples, and it is operated by machine or tractor. Some power is used. So what are different tools for sampling? Soil auger can be taken, or a spade can be taken. Auger is the name of a sampling tool that you will see now. A clean bucket or a tray or a clean cloth for mixing the soil and subsampling. Polythene or cloth bag of specific size. Coping pencil, pen pencil required, and soil sample information sheet or label that you will put on the sample for identification. You can see different kind of tools, like one was cutaway. Cutaway can be like this cutaway, and then this is tube auger. This one is tube auger. This is normal auger, and this is screw, SCREW, screw auger, and this is core sampler. And this you can see, this is bucket, spade. Spade can be of different type. When using a spade, now in India, sometimes farmers, they do not have auger. In soil testing lab, like our lab, many labs, all labs, we have variety of augers that can be used to take sample, but at farmers level, it is mostly spade. And spade is as effective as your auger, etc. So no problem with the spade. Make a V-shaped cut in the soil, and it should be 0 to 15 centimeter deep in the center, and take a 1 centimeter slice from the smooth side or from the both side. You need to take 1 centimeter, 1.5 centimeter thickness of the slice. You cut from both sides, slice up to the lower end. Trim sides with a sharp blade or a pen knife, or even a spade can be used, leaving a 2 centimeter strip. Collect this into a clean bucket. So from both sides of this V-shaped structure, you take the samples. Take a number of such samples to make a composite sample. So you can take from different places. So you can see it more clearly here. If you can see, you can use, this is your field, and this is how zigzag fashion you wish to take the samples. You can have the spade may be of any type, Indian type or whatever it is. And you just make a V-shaped cut on both sides of the soil. And you make like this, and the height in the central should be 15 centimeter. And from both sides, left side and right side, you take a slice of the soil like this. You can take from both sides 1 centimeter, 1 to 2 centimeter, cut, and then put it in the bucket, mix it. While mixing, your hand should be having gloves. They should not be dirty hands. And then you got this sample, and you can divide it into 4 part or 2 part. Likewise, you can divide, and when your about 500 gram is left, you can consider it as a representative sample. So this is how this can be divided into 4 parts. So these 2 parts can be thrown away, and remaining 2 parts can be mixed, and then they can be further divided into 4, and 2 can be taken, and 2 can be removed. So this is how sampling can be taken. Now processing comes. Now you are preparing it for analysis. You have done the sampling. So drying is number 1. Number 2, sieving, grinding, mixing, and then storing or going for analysis. So you can see how we can do. It is air dried. Most of the time, the air drying is done. Oven drying is not done. If you dry in the oven, sample will lose a lot of nitrogen and sulfur. So, and sometimes drying can be done for the sample in case you are using wet sample for analysis. Sometimes wet samples or fresh samples without air drying are to be used for analysis. For example, if you want to estimate ammonical nitrogen or nitrate nitrogen in the soil, then you need to take wet samples. Otherwise, if you take dry samples, then conversion of nitrate and ammonium will change. The data of ammonium and nitrate will change. It can increase during the storage or it can decrease. So therefore, in ammonical and nitrate nitrogen analysis, you always need to take fresh sample and analyze and do the analysis in the fresh sample. However, a duplicate sample of the same can be kept in the oven to determine the moisture. And once you have determined the moisture, you can report the ammonical and nitrate nitrogen based upon the oven dry weight of the soil. So samples are generally, or rather most of the time, air dried at 25 to 30 degrees centigrade. Relative humidity can be 20 to 60%. And then they can be stored or they can be directly taken for grinding. But for certain determination like ammonium nitrate, exchangeable K, acid extractable P, and ferrous iron, fresh samples are used. You cannot wait for drying. Results expressed on oven dry basis by determination of moisture percentage by drying a small sample in an oven. Duplicate sample. After this drying, then comes the sieving. It is not grinding, it is sieving. So sieving field moist samples prior to drying can be made to pass. Means initially when you bring the soil sample, sieving can be done at that stage also. About 4 mesh per inch. This 6 millimeter sieve can be taken, which will have 4 mesh per inch. 1 inch means 2.5 centimeter approximately. So in 2.5 centimeter, you will have 4 holes. So therefore, it is called 6 mm sieve. 6 mm sieve, about 4 mesh. Yeah, in 1 inch or 2.54 centimeter, you will have 4 holes. And then fresh sample can be rubbed by fingers, and this practice seems of much advantage in case of heavy soils. So what is there? Certain stones may be there that can be cleaned at this stage. Soils in the right moisture condition can even be passed through a 2 millimeter sieve, that is 20 mesh per inch. The common practice of sieving a portion of the gross sample through a 2 mm sieve and discarding the rest is not desirable, as it increases the concentration of most elements involved in soil fertility. When the gravels in the soil exceed 2%, limit of over a 2 mm sieve, their exact percentage should be recorded. Now, after sieving, now grinding can be done. Grinding can be done manually by hand also or by machine also. Grinder is available. A roller rubber pestle in an agate mortar or a motorized grinder is commonly employed. Grinder is available. Crushing of the gravel and primary sand particles should be avoided because they have already been cleaned. They should be cleaned. For heavy soils, as indicated earlier, it is better to pass these through a 2 mm sieve before allowing them to get completely air dried. Now you can see these are the grinding options. One is by grinder, agate grinder, and one is by manual method, pestle and mortar. This is pestle and this is mortar. Now, then comes the mixing again. Mixing thoroughly by rolling procedure. Place the dried ground and sieved sample on a piece of cloth. Grasp the opposite corners and then holding one corner down, pull the other corner across the sample. You need to divide again because this whole 500 cannot be used. Now process is repeated back in the reverse direction. Use the other 2 corners and roll the soil. Means on cloth, you can also mix and divide it into half like this. Then you can store the soil in paper cartons. soil sample box using a polythene bag as an inner lining. Label the carton properly, giving cultivators or experimental name, plot number, etc. Means you need to write the identification details of the sample. Now, precautions in soil sampling means when you are taking samples in the field, what precautions you should keep in mind. So do not sample unusual area that is unevenly fertilized. What happens sometimes when you put organic manure into the soil, they make a small heap in the field, and after few days, say after 10-15 days, they will spread it in the field. So where the heap was made, soil will be more fertile than the rest of the soil. So care should be taken that from such area, sampling should be avoided. So site of previous compost pile and other unrepresentative sites. For a soft and moist soil, the tube auger or spade is considered satisfactory. Tube auger or spade. For harder soil, a screw auger may be more convenient. Where crops have been planted in rows, collect samples from middle of the rows so as to avoid the area where fertilizer has been banded place. So this is actually taking the sampling from the middle rows is not required for soil sampling for the farmer, but it can be done in the research part. Sometimes students do it, that how much is the depletion in soil fertility after 20 days, after 50 days, and so on. Avoid any type of contamination at all stages. Soil samples should never be kept in the store with fertilizer materials. So there is possibility of contamination. Then all kind of contamination to the sample should be avoided. Before putting soil sample in bag, they should be examined for cleanliness. If the soil is to be analyzed for trace elements, this is important, dear student. If it is to be analyzed for micronutrients like copper, zinc, and brass, copper, zinc, and brass must be avoided during grinding and handling. So what happens sometimes, your grinding machine may contain some copper material or may contain iron material or some brass material. So those kind of things should be avoided. Otherwise, they can contaminate your soil. Now, after your preparation of the sample for analysis, your sample is ready. You can store it or you can use it for analysis. So different, in the beginning, you need to extract the nutrients. I already discussed this extraction of nutrients in some of the lecture. For available nitrogen, for available P, available potassium, different extractants are used. So first you need to extract the nutrient from the soil sample, and then you analyze them by different methods, and then interpretation of soil analysis data. Interpretation I have already shown you in soil health card while we were dealing about analysis of nitrogen, organic matter, and so on. So that time we have seen interpretation. So whatever results we have obtained, we need to compare with the standard charts or standard data which is available for different nutrients, very high, high, medium, low, very low, something. So interpretation is required, and you can do it, already covered this interpretation part. Now we go to the plant chemical analysis. After soil analysis, now we go to plant chemical analysis. So if you remember, in soil analysis, there were different steps, but the major and topmost step was collection of representative sample. And second thing is that the sample should not be contaminated during its sampling or during its transport or even during analysis. Then your results will not be true representative of the soil sample. Now we go to the plant chemical analysis. So many times plant chemical analysis is required. We discussed in the soil fertility evaluation why it is required plant chemical analysis, and particularly quantitative analysis, where we calculate the quantity of nutrient on percentage basis or in other units. So plant analysis, sometimes referred to as leaf analysis also, is the technique for determination or determining the element content of tissue of a particular plant part. So you can take the tissue, you can take the whole plant, you can take just stem, whatever you want, but they are plant parts. Plant analysis can play a major role when diagnosing mineral nutrition problems. Some particular nutrient is in deficiency or in excess, then it can be very, very useful, whether for research purpose or problem solving. So this is also important. So overall, there can be many purposes of plant chemical analysis. One is to know the deficiency or toxicity, or see whether it is having sufficient level or insufficient level of a particular nutrient. And also you can correlate this with the deficiency symptoms. And then nutrient recommendation also, it can partly help in the nutrient recommendation and also in the uptake of the nutrients. You need to analyze, particularly during tilling or during any growth stage of any plant. If you want to know how much nutrient has been accumulated, then you need to analyze it. And you also know the dry matter and you can know the uptake. Similarly, at harvest, you separate your harvest biomass into grain and straw. And then you can know the concentration in grain and straw. And then you can easily work out the nutrient uptake. So it is also required for knowing the uptake of the nutrients. Now, what can be done in soil chemical, plant chemical analysis? Almost same steps are there, same steps. You can see sampling, sample preparation or processing, and then elemental analysis, and then interpretation. So these are 4 steps. Sampling of plants. A research worker in soil fertility may be interested in finding out the nutrient uptake by crop at different growth stages. The stages at which nutrient uptake are to be studied will vary with the different crops, but for most times, people like to know the uptake during flowering stage, particularly in annual crops. Annual crops, the students and scientists are mostly interested to know the status of nutrient, particularly at the flowering stage. So normally there is a high requirement of nutrients, particularly certain nutrients at the time of flowering. So therefore, they would like to see that what is the nutrient status at flowering. One may also be interested in collecting a plant sample when an abnormality is observed. Some deficiency symptom is there, but you don't know what is the cause of deficiency or toxicity. Then you can analyze and see the results. In such cases, no time should be lost. If you think that it is deficiency or toxicity, immediately you can analyze it. It can be done in 1 or 2 days' time, because here you need not to go for air drying of the soil sample, like in case of soil analysis. Here you can go for oven drying of the sample. That can be done in 24 hours, and then immediately you can analyze your sample. So it may also be mentioned here that in some cases only the affected plants are to be sampled if you see deficiency or toxicity. Now, what area of the field can be representative of one plant sample? So for research project or students, each plot receiving differential fertilizer or some other treatment is a separate unit and is to be sampled separately. Some of you might have done research on fertilizer, and you have different treatments. Somewhere you have 0, 30, 60, 90 different levels or sources. So from each plant, you need to take a sample. For general advisory purpose, one sample would be enough if you do not have some treatments like research, etc. One should, however, take care that the sample comes from a field or part there of having a uniform stand. The plant population should be uniform. You should try to take sample from the uniform plant stand. Somewhere you get less plant, very high plant, so that will not be real representative. In a field in patchy and differences in stand and plant growth are obvious, such area should be avoided. Means, or they can be sampled separately. Sampling techniques, the person collecting the sample should travel across the rows in the center of the plot. In small plots, every alternate row, and in big fields. So that will be depending upon the interest of the researcher, what he exactly wants to sample. For a small experimental plot, say about 100 meters square, about 10 plants or 10 tissues would serve the purpose. But for bigger fields, you need about 25 to 50 plants or tissues. But sample should be representative, tissue to be sampled. So it again depends upon the need of the researcher. If you want to know uptake in the whole shoot, then you need to harvest the above ground biomass. If you want to know the critical concentration in petiole, you need to take petiole, or you need to take leaf blade, or sometimes stem, or sometimes the shoot can be separated into the leaves and the stem. So it depends upon nature, or the demand or requirement of the researcher, that what part to be sampled. Now, next comes now you have sampled the plant sample. Now, what can be done on this? Initial handling, decontamination is important, moisture removal, particle size distribution, and organic matter destruction. Initial handling, what care should be taken? Then cleaning of the sample is necessary, washing, because it may have some dust particle and some undesirable material that is required to be removed. Otherwise, that material may contain some nutrients and it can overestimate your nutrient content. Moisture removal is next stage, then grinding of the plant sample, then ashing or extraction of nutrients by dry ashing or wet ashing. So fresh plant sample is perishable. It is best to transport plant sample in a clean paper. Avoid the contamination during the transport and keeping it in the lab. Then you should keep it in the tissue at a cool temperature and maintaining the integrity of the collected sample. Decontamination, washing is very, very important. Cleaning is important. Plant tissue may have dust, soil particle, or foliar materials. They are required to be decontaminated. Only fresh, fully turgid plant tissue can be subjected to a decontamination procedure. Decontamination can be done by washing with a double distilled water, can be used for washing of the sample, or sometimes even ionized kind of water, people say. Sometimes deionized water can be used. Sometimes mechanical wiping or brushing may be sufficient to remove large soil particles. Sometimes you can use detergent powder. 0.1 to 0.3% phosphorus-free detergent solution can be used. But after that, you need to clean again by the pure water to remove the detergent completely. So one detergent is TPOL. TPOL can be used 0.1% and hydrochloric acid at 0.1 molarity, then followed the rinsing of pure water. So this is how we can decontaminate the plant sample. And then for moisture removal, you can put it at 80 degrees centigrade temperature for period till it gets the constant weight. So this is, I think, not correct. I think this is 65 degrees centigrade. So please consider it as 65 degrees centigrade for 24 hours. In India, we use 65 degrees centigrade temperature for drying the plant samples for 24 hours or till it reaches the constant weight. So now drying is done. Now you can go for grinding of the sample. So for grinding, different grinders are available, different machines are available. But here also, if you are doing analysis of micronutrient like copper, zinc, iron, then your grinder should not have these elements in them. Otherwise, they will come to the soil sample. So some options are available for this. You can find certain grinders which do not have copper, zinc, or iron, or brass fitting, etc. So they are expensive, but they are available. So then you can, after grinding, your sample is ready. You can do the weighing of the sample, and then you can go for, these are different grinders, grinding machine, and sieving can be done. And then your sample is, you can go for dry ashing. Dry ashing, I discussed, it is destruction of organic material at high temperature. You remove carbon, hydrogen, and oxygen by dry ashing, and then the nutrients are left. And then you can analyze the concentration of nutrient, and you already know weight of the plant sample. Therefore, crucibles are used. Mostly quartz crucibles are used, made up of quartz. Crucibles are small cups having 100 ml or 50 ml or say 10 ml. Different sizes of volumes are there. So this is done, and then wet etching can be done with the acids, diacid mixture, triacid mixtures. So digestion in a mixture of nitric acid and HClO4 can be done. Other acid digestion in a mixture of HNO3, 30% H2. So number of acid combinations are available. Unfortunately, you are not able to do it in the lab. That is the biggest problem with you. And I have my sympathies with you that you are not able to see it. Learning by doing is really good for practical purposes. And then you will do follow some procedures, laboratory, elemental analysis, and then interpretation. Interpretation based upon the concentration of different nutrients. You can see whether it is critical or above the critical. Is it sufficient, insufficient? So variety of interpretation you can make from the results obtained in the analysis. So any question from this practical? This is a kind of practical where preparation of soil sample and plant sample was discussed. So we go to the last link,